Welcome, my colleague and my students, in this new course about ELISA application in poultry industry in diagnosis of diseases, either viral or bacterial. This is some basics about the ELISA and some introductory knowledge we should know about antigen. Antigen, we can say any foreign substance that elicits an immune response, the production of specific antibody molecule when introduced into the tissues of the susceptible animal and is capable of combining with the specific antibodies formed antigen. There are different type of antigen. Antigen are generally of high molecular weight. This is a characteristic for this antigen, high molecular weight, commonly protein polysaccharide, give strong immune response. We say it's strongly immunogenic. The small site of an antigen. Here we can see the antigen, either viral or bacterial. This antigen, we can say this is epitopes or parts of the antigen which is immunogenic. This is stimulate the immune response for the body. The small site of an antigen to which an antibody binds is called epitope. We call these parts epitopes. Most common antigens are virus or bacteria. In our discussion about diseases in poultry, antibody, another definition. This is we speak now about the antibody. Antibody is immunoglobulin capable of a specific combination with an antigen that cause its production in susceptible animal. Antibodies are produced in response to an invasion of foreign molecule antigen in the body. We speak about antigen antibody. Antibodies exist as one or more copies of Y-shaped units. In a cartoon or in the model of the antibodies under the electron microscope, it looks like Y-shaped. This is we call this small chain and this heavy chain, and this is discussed in full detail in immunology and in another course about the fundamentals of ELISA. You can see this course it's one of my courses, and also in immunology course step by step. We now discuss some ELISA types or the difference between blocking ELISA and the indirect ELISA test. This is important in choosing the type of ELISA we use. A broad range of antibody variant will bind to the plate. We use a plate, a multi-channel pipette. We use some kits. We will discuss this. The plate is coated by antibodies. These antibodies are variant in its ability to bind to the plate. The conjugate is antibody of anti-species IgG and conjugate with bind to all already bound antibodies. We use antibody, but it's conjugated or binded with anti-species. A bound antibody highly sensitive, but a small possibility to have non-specific antibody binding to the plate, resulting in false positive results. We use only specific species depending upon which conjugate is used in the test. The amount of bound and conjugate is proportional to the antibody level in the sample. quantitative result can be calculated as sample positive ratio, and we will speak about the calculation in full detail. Here, the indirect ELISA, you imagine that this is the plate and this is one of the plate, this one here. There we conjugate, we coat the plate with antigens, and here the antibodies, here the positive result, here the negative result. Here antigen, the plate is coated with antigen. We add sample, we wash by washer or manually, and add the conjugated antibody with a chromogenic material or enzyme, like horseradish peroxidase. After that, the technique is called enzyme linked immunosorbent assay. Enzyme linked immunosorbent assay. This is from this expression, enzyme linked. Here the antibody is conjugated with enzyme. We wash every time we wash to remove the unspecific antibodies or unspecific protein. After washing, we add substrate. This is substrate react with the enzyme to produce a color, and this color can be detected by a reader, and the intensity of color is referred to the amount of this antibody. Simply, we read optical density at appropriate wavelengths. We use different filter on the reader and use the optical density to refer to the amount of antibodies bound to the antigen. Here in negative result, no antibodies found here and no color produced. This is negative result. This is the indirect ELISA. In competitive ELISA, a reverse occur. A broad range of antibody variant will bind to the plate. The conjugate is monoclonal antibody, specific antibody, which will bind to only a specific epitope on the coated plate. If this species is not already occupied by an antibody form, the sample in the high specific, it doesn't matter if any non-specific bind has happened to other parts of the plate, as this will not be detected by the conjugate. This blocking ELISA, competitive ELISA, yes, no. There is the antibodies, there is no antibodies. This is indirect proportional to the antibody level in the sample. The more antibodies present in the sample, the less color developed in the test wells. There is a reverse for the indirect ELISA, non-species dependent, as the conjugate bind directly to the antigen on the plate. Here is, we can imagine this antigen, this the added antibody, the specific antibody. We need to discuss the discover. We wash every time. If there is antibody, this is a positive result, light color or clear color. Here a negative result, the color is more intense, more dense. The conjugated antibodies bind to the antigen. Here the conjugated antibody not bind to the antigen, only the specific antibody bind to antigen and prevent this antibody, conjugate antibody, from binding to the antigen. It's competitive. It's a competition between the specific antibodies and the conjugated antibodies. reverse and reverse for the indirect ELISA. Thank you.